My name is Charvanaa. I'm a master's candidate from the EAS department, and my project focuses on investigating carbon, nutrient, and microbial pools beneath the Hayate Glacier. Oops. Sorry, I don't know why it's okay. So subglacial, glacial meltwater draining from the glacier base is called subglacial runoff. Due to its interaction with the bedrock, it can be a unique source of rock-derived nutrients and terrestrial organic matter, which is a source of carbon to downstream ecosystems. Its composition is largely shaped by components of the subglacial environment, such as basal ice and channel waters. Because these two are formed differently, they could have potentially different carbon, nutrient, and microbial properties, which remains to be investigated. Basal ice is formed through physical interactions between the bedrock and the glacier, whereas channel waters form when surface water drains to the glacier base and gets into contact with the bedrock. So the goal of this study is to determine whether different subglacial sub-environments can be distinct nutrient, carbon, and microbial pools. To answer this question, we collected basal ice samples, channel water samples, and a surface ice sample from Sverdrup Glacier in the Canadian High Arctic. First, we looked at whether dissolved organic matter composition was distinct between different subglacial sub-environments at the molecular level. Statistical analysis and this NMDS plot here shows significant difference in DOM composition at the molecular level within the subglacial environment. From these Van Crevelen plots here, where each dot represents a unique compound and different regions represent different classes of compounds, we see that the significant difference is largely driven by the higher molecular diversity we see in channel water than in basal ice. We then compared major nutrient concentrations between different subglacial sub-environments. We saw higher total phosphorus concentrations in basal ice, which largely comes from the bedrock. However, only a small fraction was bioavailable and is actually comparable to channel water concentrations. Channel water samples had higher nitrate concentration than basal ice, and this is likely from nitrification of surface ammonium or mineralization and nitrification of subglacial and surface organic matter. Finally, we compared live and dead and live-only bacterial abundances between channel water and basal ice. Live cell counts were mostly lower, were lower across all samples than live dead cell counts. With basal ice, however, we see that it may have acquired additional microbial cells from the underlying bedrock, which then got damaged the process, which explains this large decline that we're seeing here. Next steps are to characterize a subglacial microbial community composition differences. This study hopes to provide an understanding of subglacial processes affecting the composition of subglacial runoff. With increasing global temperatures, subglacial fluxes are likely to increase, as might the export of these glacial materials to downstream ecosystem. So understanding these processes on subglacial runoff composition could provide insight on the ecological function and impact of the subglacial materials that are exported to downstream ecosystem. With this, I'd like to thank my supervisors, collaborators, lab members, and my department for their ongoing support. Thank you for listening.